lecture. This lectureship has started in 2014 in occasion of the 50th anniversary of the Medical Genetics Clinic, and has the name of our first medical director. And now I'm introducing the Dean Hemelgarn, who will introduce our speaker directly. Thank you.
Thank you very much, Joanna. I just want to begin by acknowledging that the University of Alberta is located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. I also want to start off by acknowledging WICRI's foundation partners, the Stollery Children's Hospital Foundation and the Alberta Women's Health Foundation, for making this event possible. And I have the great pleasure now of introducing our guest, Dr. Gary Cutting, who's a professor of pediatrics and medicine in the McKissick-Nathans Institute of Genetic Medicine at Johns Hopkins University of Medicine. He's also the Atina Healthcare Professor of Medical Genetics at Johns Hopkins and Medical Director of the DNA Diagnostic Laboratory at Johns Hopkins Genomics. Dr. Cutting has numerous awards in the past, including the recipient of the Paul St. Agnes Distinguished Scientific Achievement Award from the Cystic Fibrosis Foundation and a Merit Award from the NIH. He's published more than 160 peer-reviewed articles and was elected to the Society of Pediatric Research, the American Society of Clinical Investigation, and the Association of American Physicians. Dr. Cutting's primary interests lie in the interpretation of DNA variation and their effect upon human phenotypes. His lab focuses on the effect of common and rare variants in the CFTR gene that causes a single gene disorder, cystic fibrosis. Finally, as editor of the journal Human Mutation, Dr. Cutting oversees the review and publication of manuscripts reporting the mechanism, distribution, and phenotype consequences of variation in our genomes. It gives me great pleasure to introduce and to welcome you, Dr. Cutting. We look forward to your lecture.
Well, thank you, Dean, for that introduction. I think we're in the process of switching over here. We're getting used to this 2D world, so thank you for the invitation, Joanna, to speak. My lectures could be based on a somewhat briefer talk that I gave at the American College of Medical Genetics earlier this year. I can't wait till we get back to the 3D world. It's a little bit more difficult giving talks on here, so I think we're going to hold the questions to the end. I don't usually mind interruptions, but I guess the format is to have them at the end. So, but I'd be more than happy to answer questions as we go forward. So we're going to have to go to the share screen, right back again to mine, and okay, here we go. So the title of my lecture is Medical Genetics and the Functional Interpretation of DNA Variants, and I'm going to draw heavily on our work in CF, which we have focused on since the cloning of the gene back in 1989. I have no, disclosures other than my funding from the NIDDK and USCF Foundation. I do want to mention, as was said earlier, that this is the Peter Bowen Lectureship, and I did take a look at Dr. Bowen and his memoriam and what he had done over his life, which was quite impressive. I noted that he had a fellowship here with Victor McKusick. A number of people over the years had worked with Victor and had been inspired by his work and went off to form other medical genetics units elsewhere. And it sounds like Dr. Bowen was an important part of the development of medical genetics in Alberta. It was impressive that he, working with Hans Zellweger, described the Zellweger syndrome, and then he went on with Conradi to describe the dysmorphic syndrome, Bowen-Conradi, and the Hutterites. Hutterites and Amish were a key interest also of Victor McKusick's, so that's an interesting connection. I also understand he worked with the Hereditary Disease Program and coordinated with genetic counseling services. So in some ways, he was sort of ahead of his time, given how we've now have intimate interaction with our counselors, and that's been a growing and continuing expanding process over the years. And finally, perhaps not at least, he directed the cytogenetic laboratory, and as a laboratory director myself, a clinical, I know how that work really translates what we do in genetics to real useful findings in the clinical arena. And he worked on delineation of Fragile X syndrome. So I'm very honored to be giving a lectureship in the name of someone so well achieved in genetics and medical genetics. So I was asked or mentioned, it was mentioned that this would be a part of the precision medicine initiative at Alberta. So I thought I'd throw in just 2 slides of what we're doing at Hopkins. And because we actually have a precision medicine center in cystic fibrosis at Hopkins, we're one of, I think, 13 centers that brings together a variety of different approaches to diseases. I thought I'd give a slide or 2 just as an introduction to that. Our program is called InHealth, and as described here, it's a strategic initiative to advance precision medicine, and it really is across the entire university. And we also work with the Applied Physics Laboratory, which is our sort of computational colleagues, intense computational colleagues, to crunch the large data sets we have developed. And in the next slide, I'm just going to show you, it's a little bit of a public announcement here, but it's one of those inspiring things that the Hopkins, I think, offices of development put together. Just describe what we're doing, because it'll lead on to how we're thinking about precision medicine and the role that genetics and variant interpretation plays.
How do you tackle the sheer complexity of disease? How can the course of a disease vary so significantly for patients with the same?
Oh, my microphone has gone out. Sorry. Sorry about that. I'll move it up.
For patients with the same diagnosis? How do you even begin to approach these kinds of questions? The answers are right here in the data. We're just the ones who dared to look at it differently, using unique filters to identify and relate the seemingly unrelated, using not just genetics, but data from imaging, sensors, laboratory analysis, and the environment, creating new solutions, unlocking more possibilities. At Johns Hopkins, we think of this as a whole person approach to precision medicine, a powerful integration of expertise and data, breaking down the most complex diseases into patient subgroups, isolating the specific factors that determine their health outcomes, and then leveraging it all across different disciplines. This is how we look at data, in ways that allow us to be more precise, more scalable, more customized to specific needs of patients. This is what we are bringing to the world, a new world of insights, solutions, capabilities, and yes, breakthroughs.
So I know that was relatively commercial and advertising for Hopkins, but I think it contains the essence of what a lot of people are envisioning as the potential precision medicine. And they do emphasize it's not just genetics, and I think that's very important. Other data sources are extremely relevant, and they're going to be, as I will show, important in the interpretation of DNA variants, because the big question shown here in this nice graphic, borrowed from the National Health Service in the UK, was which DNA variants cause disease, given we have 3 billion nucleotides and many millions of variants. We're struggling to make those decisions, particularly when they occur in genes associated with disease. And I want to emphasize it's the variants in those genes that cause disease, because some variants do cause disease and some don't. It's not genes that cause disease, it's the DNA variants that cause disease. So their interpretation is essential to what we do in genetics, and that's an essential contribution that we make to this process we call precision medicine. So I focused my work on CF, as noted earlier, for those who, just to give a little bit of background, CF is one of the most common life-shortening single gene disorders. Affects about 30,000 people in the United States and 70,000 worldwide. That might be an underestimate from our more recent data. Certainly has quite a bit of morbidity and mortality associated, even though we now have modulators out, which may change the face of CF considerably. Currently, we're still seeing quite a bit of mortality of individuals in their 30s and 40s. These are just data from the US CF population, and it's looking at the number of individuals and the cumulative percentage, and you can see that we have a loss, unfortunately, of life over the years, and particularly in the middle-aged years. And of course, what we want to correct this to the same as national averages, where people don't have a chronic disease such as CF. So it's a serious disease, and out of the single gene disorders, it's one of the most common. There are over 2,000 variants in this gene that are responsible for the in CFTR, that's the cystic fibrosis transmembrane conductance regulator, and that's the gene responsible for this disorder. So in annotating variants, we started back around about 12, 13 years ago, initially to characterize variants for the purpose of interpreting newborn screens that were positive. But it sort of grew into a larger project over time, particularly when drugs came out that were variant specific. And we developed an annotation process. And it really combines data from various sources. First of all, we populate a database from all over the world. These are individuals with cystic fibrosis, and they've been contributed from in country or region-specific databases. And we have 89,000 individuals almost in this database from 41 countries. We've selected from this information variants that are reported in 3 or more alleles. And then we've used a 3-pronged approach to analyze disease or assess disease liability. And then we report it on the website. This is the 3-pronged approach, and it combines clinical analysis, functional testing, and population and penetrant studies. And together, that's how we assess whether a variant is disease-causing or not. So it's more than just the genetics. You do need, of course, the clinical information, and you do need other types of information, such as population data. So we published in around this process in 2013, and a couple of years later, Sue Richards, working with a very good committee within combined between the American College of Medical Genetics and the pathology group, the molecular pathology group, put together guidelines using the same elements, basically, to interpret DNA variants. Here is a diagram sort of exhibiting what their approach was. So if we just take the variants themselves and we sort of look at how many end up plotting at one end of the spectrum, the pathogenic or likely pathogenic spectrum versus the benign or likely benign, that's a subset, a smaller subset of variants than those that sit in the gray zone, the variants of uncertain significance. And this is, I think, for many people in the audience, the real challenge we have in genetics is those VUSs. And the committee, headed up by Sue, put together the following approaches and a whole schema of interpreting these variants using population data, functional data, both of variant type and then from experimental studies, clinical observations, and then computational. So as you can see, sort of similar to what we had been doing with CF, they included computational data, which we have found in varying levels of usefulness, although it has a great deal of promise, and I think it's becoming more and more effective as we develop more data sets and use more approaches. So using this information, you can see we can combine it and then make a better approximation of whether something is disease-causing or not. Population data helps us assign whether something is benign but not so helpful for likely pathogenic or pathogenic. Clinical observations can help across the spectrum, as can functional studies. And so for many variants, we can assign them pretty quickly, except for really one class. Ends up being the real headache, and that's the missense variants. If you change an amino acid in a protein, it is really difficult to assess whether or not it's going to be disease-causing, as opposed to something that might introduce a new premature termination codon, like a nonsense mutation, which is much easier to interpret as likely causing disease or likely pathogenic. So we really do need functional and experimental studies to help interpret those missense variants. And so a committee was convened by the ACMP and ACMG and AMP to ask what criteria do we really need to interpret these variants on a functional level. And they really came up with 4, I think, quite useful principles. So first, if you're working on disorder, you need to define the disease mechanism. You need to evaluate the applicability of the general classes of assays that are used in the field, evaluate the validity of specific instances of those assays as they apply to that disorder you're working on, and then apply that evidence to individual interpretation. Probably easier said than done in many cases, but at least this was a framework, and I'll show how this framework applies to what we've done in CF. But I do believe it's generalizable, and we're starting to see that kind of generalizability with a project in the United States called ClinGen, and a variety of different disorders now have committees that are considering applying these criteria to functional assessments of variance. Now, it was worked out by Sean Taptigian on the committee using a Bayesian approach that you need at least 11 or so pathogenic and benign variants, or a mixture of the two, to reach a moderate level of evidence if you hadn't had a rigorous statistical analysis. I think the takeaway from that point is you can't just do this kind of study on 1 or 2 variants. You really need to do at least 10 or so and study them to get a feel for what really is going to be deleterious or not deleterious. So I think that was that point. So how does this sort of apply, and how do we work this through with cystic fibrosis? Well, first, disease mechanism. We are fortunate in CF to have a very good understanding of disease mechanism. Here is a picture of the CFTR gene, protein, I'm sorry. It is inserted into the cell membrane. There are 2 transmembrane domains shown in purple. There's an ATP binding region shown in yellow, and this regulatory domain. It is activated by ATP binding and then phosphorylated in the R domain, and that's what opens up this protein, which is actually an ion channel, and it specifically conducts chloride. So in CF, the loss of that chloride transport results in dehydration of surface liquids, resulting in the obstruction of airways and ducts. So airways in the lungs and ducts in the pancreas. And so the primary disease in CF is pulmonary disease, and pancreatic disease manifest by an inability to digest foods and unfortunately eventually also diabetes. So loss of CFTR function may occur by a variety of molecular mechanisms, and many have been explained. Ones that affect transcription of RNA, splicing of RNA, folding of the nascent protein, and then stability of the protein to the cell surface. All of these variants that alter CFTR function by these mechanisms affect the quantity of CFTR. Then there's a series of other variants that alter the activity of this protein or its conductance. That's the movement of the ions through this ion channel. And those are called functional variants. So using that as a framework, we'll go forward to applying some of those principles that the committee have put together. So first, let's think about the distribution of CFTR variants by predicted effect. So the source here is the CFTR2 database, and in that database there are 1,749 variants. Again, these are individuals who have cystic fibrosis. They're in the database. And what's interesting here is that the majority, 55%, are actually predicted to affect RNA processing. And this is not actually that unusual for many recessive loss of function disorders. Missense variants are the next largest category, and they present that substantial challenge in interpretation. There's a variety of other variant groups, the complex ones and the synonymous or unknowns. Of those RNA processing, you can break those down to the ones that you would expect to see in this category: frame shift variants, nonsense, canonical splice, other types of splice, deletions, duplications, and promoter. And all of these, when their effects, will alter the amount of RNA and finally the amount of protein in cells. So they are going to affect quantity. So what methods, if you're going to consider in that second step, what assays are you going to apply to evaluate quantity? I'm going to focus on RNA transcripts. Where do we start? Well, you could perform RNA on samples directly from patients. However, for a variety of diseases, even including CF, the appropriate cell type can be difficult to access. If you're working on a gene that's expressed primarily in the liver, is it possible to evaluate effects in peripheral tissue, skin, or blood cells, or must you actually access liver tissue to look at effects on splicing? Particularly splicing effects differ per tissue type. It's very hard to sample, to get samples if there's a very small number of individuals bearing certain mutations, and there are a lot of rare variants, not only in CFTR, but in all these diseases. So actually getting individuals that carry these variants is a challenge. And of course, for many disorders, and CFTR is no exception, there are a large number of variants to be tested. So it is a challenge to go with direct samples, but directly with samples from the patients, but very valuable when you can obtain. There are in silico tools for prediction, but there's not great concordance amongst the different programs. Although, as I mentioned earlier, these tools are improving progressively. And there are problems with variants well outside of the consensus splice sites, for example. So again, the tools are developing, but are not really ready for prime time, though they are a useful adjunct to your work. Hybrid mini genes have been used, and these contain exons from a different gene, and you place your exon containing your variant or your region containing the variant into that context, and you evaluate the degree of splicing. And the problem is it's being tested in a non-native context, and there's no information on actually the effect of the missplicing on the target protein. So we and others have focused on an expression mini gene, which has been developed over the past 8 years or so. A colleague of mine here at Hopkins, Neeraj Sharma, has worked extensively on this, and there's several colleagues that we have collaborated in Europe who we've collaborated with in the CF field and others in other fields, particularly retinal diseases. And this is an interesting system because the expression mini gene contains the entire coding sequence of the gene of interest, and you insert into it introns and exons. You usually can't put the entire intron in, so you put in a truncated sequence, about 200 to 300 base pairs flanking the exons. And that sequence generally contains all of the signals you need. Now, I'll acknowledge that more distant splice repressors and enhancers will not be included. But for the vast majority of the cases, you will be including the key sequences for splicing. And the key thing here is because you have the entire coding sequence, you can not only look at the effect on splicing, you can look at the effect on protein levels, protein stability, and protein function. All can be done in one system. So here's one of the first ones we developed. This is an expression mini gene, contains here, driven by the CMV promoter. It contains exons 1 through 11. These are just the coding sequence, the exons, and then exons 13 through 27. And sitting here is exon 12, flanked by intronic sequence. Very simple EMG, but now you can look at the effect of splice variants that sit in the splice donor site, splice acceptor site. Here you can look at the same things here and here, and you can make more complex EMGs. And we've now constructed ones that can have 4, 5, 6, I think we're up to 8 exons at one time. And in fact, we rarely, if ever, look at a single exon time. We almost always, if we're going to study the effective variant, we will have sequence upstream and downstream from at least one exon and its flanking introns. Upstream and downstream frequently we'll do more because sometimes the pattern of missplicing involves more than one exon. Sorry. So here's an example studied by Neeraj working with our group, in which we looked at a series of different nucleotide variants around the same splice acceptor, and each of them resulted in a somewhat different predicted effects. In some cases, it was predicted we would create a new AG, so the splicing might occur again through this new AG. Same thing here and here. The question is, would there be missplicing, or would there be appropriate splicing? And if there's appropriate splicing, would there be a stable protein made? So if you looked at the RNA level, and here's the different variants shown here, you can see in certain cases like this -1 and -2, you get misspliced transcript. This is the smaller transcript. This is the wild type size transcript. And same thing with -3. But then there's no aberrant transcript seen with these at -8 and -9. Is that really wild type or not? Well, if you sequence it, you find out, in fact, it's misspliced and a new splice site is used. However, the resulting transcript does not encode a functional or stable protein because this is a Western blot that can be done simultaneously. This is the control protein. And here you see wild type CFTR protein made from expression, the exact same expression mini gene. And here's a different cDNA, just another control. Anti-CFTR antibodies used here. And you can see, except for a faint band here for the -2, no protein is made. In fact, here's where we, over several experiments, quantified it. And so if you combine the RNA data with the protein data, you get some answers right off the bat. And you could argue that every single one of these variants now, because they produce no protein, via a variety of different mechanisms, I might say, they are going to be deleterious. So you could predict these would be pathogenic. Now, if it's not a splice site mutation, so I just shift gears a little bit here, and it's just a missense mutation, you also want to look at protein quantity. And again, straightforward processes like Western blotting can be used, and you can scale these up. I'm just showing a very simplified system here, just used in the lab, but there are automated systems that can do Western blots on very large numbers of plasmids and variants. And here, just an expression of the wild type protein CFTR, and then that same construct now contain the delta F508 mutation. This is the common mutation. This mutation causes misfolding of CFTR such that you don't properly glycosylate the protein, and you get this incomplete glycosylated B band. There's no C band, which is the mature glycosylated form. So the Western blot, in this case, immediately shows a loss of quantity of wild type protein here and the presence of aberrantly processed protein. In the case of this variant, G551D, another disease-causing variant, well worked out, this does not affect the processing of CFTR. There's plenty of properly processed protein at the cell surface. So in this case, you're going to have to do a functional study to determine what the effect is. In the case of delta F, you can pretty much predict it's going to be disease-causing due to the absence of the protein. So a simple Western blot can tell you quite a bit about what is going on, might be the effects of a variant on the quantity of protein being made if it's a missense variant, for example, or an in-frame deletion like F508 del. This was transient expression. You can also use stable expression, and we'll show you how stable expression has been used extensively for us, because when you do stable expression and you create a G551D cell line with stable expression and you use the right cell types, you actually can study function in that same cell line. So one cell line used to study both protein processing and protein function. So again, I'm trying to get across the idea that you develop these systems to assay more than one effect of these variants. In the case of the wild type, I just want to point out that there's actually different levels of protein with this cell line, the stable cell line. I'm going to talk about this in a minute, but we took advantage of the fact that these are isogenic cells, and they can produce different levels of protein. So you have to, when using stable expression, you also have to look at the level of RNA expression to adjust for that. So there's nuances to all these systems when you develop them, but again, once you work them out, you can use them extensively. The system that I'm going to tell you about, we've looked at over 200 missense variants in the CFTR gene, and the company Vertex has used a very similar system. We developed that with them some years ago, and they have used that extensively to test the effect of all their modulator drugs. We have several other colleagues also using similar systems, and it allows us to compare across those different laboratories, and so we can ensure we get replication results and have confidence in the calls on these variants, because multiple labs are studying the same variants using similar systems, but in obviously different labs. Now, again, here's wild type fully processed to the C band, and here's delta F in that stable cell expression system with just immature protein, very tiny amounts of the mature proteins. So again, if you're using the stable cell expression system, same kind of result you get from the transient system, that you form no mature proteins. So you could use transient expression, you could use stable expression, advantages to each of these. Many would argue this is a much more rapid approach to assessing protein processing and protein amounts. Certainly would agree. If you've got to do functional studies after doing this kind, say, well, it's processed protein, then this type of stable expression system is a good one to use. There are other very effects to consider. I'm just going over the ones that affect the amount of protein and so forth, but there are combinations of variants, complex alleles, as we call them, where there's more than one variant in the same gene, and you've got to assess the effect of these variants one upon the other. That gets it more complicated. Cryptic splicing can occur outside of the canonical signal, so you have to find ways to assess those. And then you can have variants, not a large number reported yet, but they can alter the translation rate because the change of the nucleotide now requires the use of an infrequent tRNA in the cell, and it slows down the process of translation, which results in problems with folding of the protein. So we're finding more and more about the effect of these variants. I'm taking a pretty simplified approach is quantity and function, but there are many other effects that we're starting to look at that we're going to have to consider as we move forward. Finally, the third step, I should say, is validity. Do these systems really, are they comparable to what happens in vivo? For example, we've looked at studies we've done on the spliced variants in CFTR. We've looked at the misspliced isoforms we've seen in our EMGs, and then we've actually got tissues from patients bearing those variants, and we've compared results. And in each of these cases, we actually do find good concordance. In the case of, I think, one of these, we have a slightly different result that we got a deletion of 53 nucleotides in exon 22. In the EMG, this was not observed in the primary. These are human nasal epithelial cells. But regardless, the key effect, which would cause this variant to have loss of function, is this retention of 40 nucleotides, and this was observed. So for for most of these variants, I think we have very good correlation between this experimental system and what we've seen in primary cells. I think we're up to about 8 different comparisons now, and this correlation is held up. But a critical step, if you're going to use a system that is in the lab and allows you to rapidly assess these variants, then you need to be sure that they are reflecting the in vivo situation. And again, looking at primary cells when you get when they're available is a key way to assess validity. And finally, you can apply this information. So, okay, you go ahead, you've got the system working, and an EMG system, for example, is the way we've gone. We transcribed or transcribe and translate these variants, and we look at whether transcripts made, partially stable transcript or stable transcript. Then we, in the same system, can look whether there is protein or not, no protein, truncated missense protein, misprocessed protein, truncated processed protein, or near full-length protein. And finally, we can interpret the effect. In the cases where no protein is made or truncated misprocessed protein, yes, we can make the call for disease causing. Again, all in one system, as I showed you before. And then in certain situations where we have truncated processed protein or near full-length protein, we can't make a determination of the effect. Is it deleterious or not? We don't know. That's where we have to do functional testing. So I have to shift now from assessing quantity to now thinking about, well, okay, functional assays. Really, functional assays are just a representation of what we think is going on in the patient. And we and others have, over the years, utilized those cell-based studies really as a way of sort of connecting everything together, running from the gene, RNA, protein, cell, tissue, organ, all the way to the patient. And I've shown you studies where we go from the gene to the protein to then in the cell system. And we like to think, and we have good evidence, that the cell-based results reflect what's going on in the patient, because we're really sort of following this paradigm of variant, the function of that variant then can be interpreted into the severity of the disease. The other great value of developing cell-based systems is to evaluate therapeutic response. If you are developing drugs and you have cell-based systems that do represent the in vivo effect of variants, they can also be used to test therapies, whether it be a genome editing therapy, a modulator therapy, or some other therapy. You now have a system developed representative of the in vivo disease for the next and probably more important step, which is finding a way to treat patients. So cell-based systems, well, what cells can you use? Well, primary cells, I've mentioned it before. They are in the appropriate context. They reflect what's going on in the individual. There you get, in the case of CFTR or whatever gene you're looking at, you get the combined contribution of all the variants in that gene, not just a single variant you're studying in the same cDNA. So that's very useful. You also get the genetic background. So you get all that information if you use primary cells. However, and I touched on this, accessioning primary cells in humans has the issues of consent, privacy, and commercialization. So those make it more challenged to take these kinds of approaches. There's also heterogeneity of the cell types you get from the patients, how they're cultured, genomic differences amongst individuals that now act to confound your results, and availability. Again, with CF, as with many other diseases, individuals with rare genotypes are, well, they're rare individuals and they're difficult to accession. And of course, trying to propagate them is also challenging. Primary cells only go a certain number of divisions, and so you have almost always a very limited supply. You can create pluripotent stem cells, and this looks like a really excellent approach, but again, there's a lot of work that's done in creating pluripotent stem cells of specific genotypes. So what about immortalized cell lines? And I sort of hinted that these could be used, and indeed they are used for a variety of different situations. But you've got to consider the species. You're going to use human, or you're going to use other species. What is the tissue source? How well differentiated are they? How are you going to express the transgene if you want to study the effect of variants in a particular gene? Are you going to do it transient, stable, integrated? Are you going to put in variants that have genes that or constructs that have one variant, or are you going to put in complex loads, or both? What about endogenous gene expression levels? Are you going to try to get it to the same level as occurs in vivo? Or are you willing to say it's okay if I have overexpression? I just want to see what the effect is on protein function. Again, if you have residual function, 1% residual function, but you overexpress it by 10-fold, you then come up with a calculation of 10% function. So how's that interpreted? So you've got to very carefully consider how your system replicates the endogenous gene expression. So this is in cap, and then the question is, when you think about the systems, which of those systems are you going to use? So I just sort of highlight again, transient expression, you're going to transfect a subset of cells, and you can transfect them at different levels. Different numbers of plasmids will get into each of the cells, and you'll have this heterogeneity of expression. That's fine if you're doing Western blots and things like that, it's fine. But if you start to do functional studies, this becomes a bit of a problem. You can try regular stable expression, but you don't know where it's integrating. So your cell line, if it is an integration method, if there's no control over copy number, so you can have some cells expressing a huge amount of your transgene, others expressing very little. So that again is a bit of a problem, because that's not what happens in most cell lines. Most cells in vivo, unless you're really working with heterogeneous cell aggregations. What we really want to do is get isogenic cells so that they have the same level of expression in each of the cells, so we can assess function in a uniform way. So this is why we went to a recombinase mediated stable expression, because you integrate your cDNA of interest into a single location in every cell. So you're making isogenic cells, and now you get much more homogeneous expression. And here is CFTR, which is fused to green fluorescent protein, and it's being expressed. I'm showing you an XZ reconstruction of cells transfected, stably expressing this fusion protein. You can see a nice green pattern, because this is GFP fused to CFTR in the apical membrane of all of these cells. And so it's a very nice uniform expression system. And then the second step you need is a good functional assay. And we are fortunate in CF, but many other disorders have good functional assays. If it's an enzyme or it's an ion channel or whatever, there are well worked out systems you can use once you've got your cell lines established. And this is the way in which we measure CFTR function. We put high chloride on one side of a monolayer of the cells that we've created. They're expressing mutant forms of CFTR. We put low chloride here, so chloride's going to move from here to here. And what we do is we clamp voltage, so we use the electrical conductance chloride as a sort of a carrier of electrical electrical charge, and we can measure that as a current change when it moves from this side to this side. And so what happens is you start recording the current over time and then stabilizes as you clamp the voltage. And then we activate CFTR with a compound forskolin, which is not overly specific for CFTR. So it's essential we then use an inhibitor, which I'm showing now. This is the current that occurs, increasing current attributable to CFTR. And you can be confident CFTR because when you apply the inhibitor, now the current decreases, and now you can measure this as the inhibition, and that's the CFTR specific current. So you have an assay system that quite precisely measures the function of the protein you're interested in. And again, there are many assays developed by our colleagues in physiology, cell biology, and so forth, and through collaboration or whatever, you can develop these systems for application for specific genes you're interested in. Here's the cell-based system we developed that has a specific integration site on chromosome 8 of these CF airway cells. So these are cells that are epithelial cells that are derived from a patient with CF. The CFTR gene itself does not express, it's undergone some evolution, and so it does not express the delta F508 mutant, which is what is the mutation in homozygosity in the donor of these cells. But we have now introduced this site into chromosome 8, and we can target that site as shown here using a recombinase. This is the recombinase plasmid. This is the targeting plasmid. You put CFTR into this location, and now these cells, which we call them flipping because this is a flip-based recombinase, can now express CFTR. These are different cell lines that are already stably expressing CFTR, and these are native cells, HBE cells, expressing CFTR. So now you can express wild-type CFTR in these systems, and you can then measure function. So here's the CF8 flip cells with no integration. Here's the integration of wild-type CFTR in these cells. And here's another cell line, which is made by transduction, viral transduction, that has many, many copies of CFTR. And what you see is huge currents. And the issue is, these are not the currents you see in native cells. They're more around this region. So these controlled expression, these integrated cell lines express at a level that's much more commensurate with the in vivo levels. So another advantage of using these controlled systems in trying to assess the effect of the variance, because as I said before, if you way overexpress a minimally functional variant, you're going to misinterpret the effect of the variant on function. Then by developing, so now to go to validity, now to actually show us how is this valid, and can we show this is valid, and how do we assess that? Well, the next step is was to develop a whole series of cell lines that are expressing different levels of CFTR RNA. And this is because cell lines, isogenic cell lines in general, just have idiosyncrasies that they express at different levels, even though they have an integration of the exact same sequence, the exact same location. There are other factors that contribute to the variability. And this was shown with pluripotent stem cell work many years ago. So I was a little surprised when I saw this result and was scratching my head, but then a colleague of mine in one of the faculty presentations said, you know, Gary, this is a well-known phenomenon, you should know about it, and sent me a series of papers that explained the stochastic occurrence of variation expression from individual loci, even in isogenic cell lines. So we took advantage of that because we had a variety of cell lines expressing different levels of RNA, and then we could correlate it with function. And so you could see a very nice correlation between the level of RNA expressed and the level of function. And this is what we call our 100% line, because this is the function we're getting when we put wild-type CFTR into the cell line, and it's expressed at different levels. So that's 100%. And then from there, you can derive 25%, 10%, and even 1% function. So a variant that doesn't produce at a certain level of RNA that produces function at this level would be about 1% function. One that was at this level would be 10% function. And in fact, to see this better, we plot this in a semi-log fashion. So we plot the log function shown here. So we can clearly see the difference between 1%, 10, 25, and 100%, because many of the variants we're going to be dealing with are going to be in this range, because less than 10% wild-type function is the threshold for full-blown CF. So pancreatic sufficient and insufficient multi-organ disease, CF, occurs at levels of function in general below 10%. So if now you've got this system validated, you can start taking a look at variants of previously well-established function. You can plot them on this and see what these are 4 different independently derived cell lines. They have different levels of RNA expression, as you can see here. And you can see that the current associated with them plots here. So they've got about 1% wild type function. Here are 3 cell lines with the same variant expressed at different levels. And you can see they very nicely plot at about the same level, about 8% of function. And this variant is known to be associated with the pancreatic sufficient form of CF. This is a somewhat milder form of the full-blown version of CF, affecting all organs, but the pancreas is not as severely affected as individuals carrying this variant. So that goes along with the idea that they have 8% or so of function. Now we have a variant that can be variably associated with CF, and in some cases causes male infertility, sometimes causes CF. The 3 cell lines here show that the function's about 20%. So now we're in this kind of a gray zone, but we can use this to then correlate with the phenotypes we see, and it makes good sense. And finally, an amino acid change at the same location as the common variant, F508. So that phenylalanine, which when deleted, we show you the results down here, has a severe effect on folding of this protein. A slight amount, a very small amount, folds it sufficiently properly to generate some function. But when you put a cysteine in there, the initial bioinformatic analysis was saying that's going to be deleterious. But actually, studies in humans, and particularly men who carry the F508 del mutation and carry in their other gene, the F508C, those males were fertile and healthy, suggesting F508C was not disease-causing. And indeed, if you study this functionally, and we are replicating functional studies that have been done by many others that show that F508C is associated with 100% wild-type function, despite what you might think right from the outset, placement of a cysteine at a key location of the protein would render it non-functional. It doesn't. So both data from the study of pedigrees, as well as clinical studies and then functional studies all combined to show F508C is a benign variant. Final application. Now you really want to apply it and see how well this works when you have studied 10 or more variants. And what I'm showing you in the dark red and light red are variants associated with CF, and they've been annotated as CF causing by a variety of other data. And I'm just now showing you the functional data. The somewhat more severe form here, the pancreatic sufficient form, is shown in the darker red, and some that are not as severe are shown in the lighter red. There are variants of varying clinical consequences where they sometimes cause CF and sometimes don't. Plotted here, we're not showing you any of the non-CF causing or indeterminates. I'm just showing the difference between these 3. Here's function plotted here, and here's lung disease severity. So as you can see, it's not a perfect correlation. It's reasonable at 0.58. It is significant. But you can see when you start plotting out function of these different variants associated with less severe CF or more severe CF, in general, there's a relatively good correlation. So 1% or so, 2% or less of function, you generally have the more severe form. Greater than 1%, but up to 10%, you get the less severe form of CF. And then above that, you start getting milder forms of CF and situations in which a subset of the CF features are present in individuals such as male infertility. Now, I assume people ask me, well, that's so well and good, Gary, but what about these why do these have low function if you told me they have the milder form of CF? Why are some people with over 50% function out here? Why are some of these discrepancies here? Relatively good function, I'll give this one here, good function, and yet supposedly more severe disease. Well, every system has its problems. I can say that to expect from any kind of functional assay or so for absolute correlation, I think is too much of a demand for the system. However, even though this might be in a higher range than I might have expected for the more severe form, it's still less than 10%. So we still have a degree of confidence that we could call it CF causing, though I can't quite explain why it's found in patients with a more severe form, even though they have 8% function. Maybe there's something we're missing in the way this protein operates in vivo, or there's some other effect that we haven't considered. But for all intents and purposes, these sort of rapid isogenic systems have proven themselves to be quite valuable. So, and that's that threshold I wanted to point out. So I'll finish here. So application of functional data to interpretation of variance. I gave you the example of CF, but I do believe the same principles can be applied. Once you define the disease mechanism for your gene or genes of interest, you think about what's the applicability of the general classes of assays. What should I use for something that's an enzyme, or so it's in a metabolic disease, or what should I use for something that is involved in a muscle disorder and so forth. And I think there's a lot of basic scientists that have been working on these systems for quite a few years and have very sophisticated assays. In fact, ones that are specific enough to be able to interpret the function of the variance when you place it in these expression systems. And then finally, at the end of the day, after you've done the validation, then you apply that evidence with 10 or more variants to your phenotypes, and you ask, do I believe what I'm seeing? Is it really interpreting the disease in the correct way? So with that, I'll finish. I want to acknowledge quite a few people over many years been working on, both within my lab, specific instances, their contributions were noted on slides. Karen Rari, I'm conflating the words here, Karen Rari and Patrick Sosny, two colleagues work with me on the CFTR2 team, as well as other colleagues. This is the International Collection of CFTR Variants. There's a missense group from various other labs, and these labs have been working with us to perform independently studies of function and RNA effects so that we can correlate results amongst different labs, collaborators at various places around the country and in other countries, and then other Hopkins faculty members. And finally, patient staff and also the clinicians who we work hand in hand with, because none of this really works if you don't have the clinical information and you don't have a close coordination, as precision medicine indicates, between all the people working on these patients, people in the basic science group, the translational scientists, the clinicians, and the clinical scientists working together to do this type of interpretation. And with that, I'll thank you and appreciate the invitation to talk.
Well, thank you very much, Dr. Cutting. This is really a fantastic overview of how we collect evidence in this time when we are working on discovering gene, but mostly in interpreting variants, because they are showering over us, and it is very important to understand how we develop that evidence. I will invite everybody to raise their hand or put their question on the chat. We actually have a first question for you already from Dr. Aitchison. Thanks for a very illuminating talk. For some of the unexpected data points, could there be effects such as epistasis operating, please?
Yeah, good point. So interaction between variants and other genes, variants even within the same gene we've seen, yes, and there could also be things that we haven't considered, methylation effects, effects on chromatin structure, that effects on translational efficiency that we don't capture. So the effectiveness of our systems for interpretation are limited by currently our understanding of all of the all the effects that variants can really have. And I think we continually get surprised by publications that we see in other genes that report very good evidence that there's a different mechanism working than expected on a particular variant. So I think we are approximating reasonably well what's going on, but we must always keep our eyes open, as pointed out here. When you have unexpected data, something really plots out of the expected range. I'll give you an example. We have another phenylalanine deletion in a critical region of the protein, and you'd think it would really be devastating protein function. It is not. And for some reason, the patients have elevated sweat chlorides in the diagnostic range for CF, even though the protein appears to be functioning at least with 50% wild type. And we've studied dozens of patients carrying this variant, and their sweat chlorides are elevated in the 80s, 90s, the same as we see in a lot of patients with CF. And they have some lung disease and other, but we can't figure. So now brings up the question is, are there certain variants which operate in a way that is tissue specific? These are missense, or it's already in this case, an in-frame deletion. So again, yep, epistasis is one of them, but I can't emphasize enough that there are probably other things that we don't capture in these systems, and it's really important that you understand what you're not capturing as much as what you are when you apply these systems of variant interpretation.
Thanks very much. Sure.
I would encourage the trainees if they have any question for Dr. Cutting.
So I do see the Gattaca comment.
Okay.
I'll get to this one. And no, I hope I just emphasized why Gattaca is a silly movie, because genetics doesn't determine all of it. There are so many other stochastic effects and other effects that occur, both known effects that we just effects that we just don't know about right now that we might call stochastic, and ones that are truly stochastic. So I think that the Gattaca is kind of overblown in its attempt to say how genetics can be so determinant of a variety of complex traits. Even in the situation of cystic fibrosis, single gene disorder, we have challenges in interpretation of the genetic changes and the phenotypes that are created. There appears to be a few other questions.
Yeah, so there is a follow-up to the Gattaca.
Yeah, excellent question.
from Martha McKinney. Thank you for an excellent and very clear talk. In the US, the FDA has accepted in vitro data to approve use of CFTR modulators in some rare mutation types. In Canada, Health Canada is still not accepting in vitro data. For patients with rare genotypes, this is probably the only way to get supporting data. Clinical trials for them are prohibitive due to cost, small numbers. CF Canada is talking with HC. Do you have any advice?
It's rather frustrating, and we have excellent collaborators in Toronto, Joanna Romans and others who work with us on the CFTR2 project, and this has been a point of frustration. I think I understand the rationale, and one could argue that in some cases the US FDA has moved ahead where we can actually, so for example, the most recent approval of variants to expand the list of variants that are eligible for these modulators included 12 that are not disease causing. Because the FDA works with the company that sells the drug, you see, and it's so, there are some complications that occur in the situation, so I can understand at least the hesitancy to move rapidly ahead. However, there's a pragmatic question that you bring up, which is with the rare variants, there really aren't any other systems available. Yes, accessing the tissues in those individuals, and we and others have been doing that. But really, if we don't use these representative systems, then there's no way to perform clinical trials for the people, for 5 people, 3 people carrying a variant. And if those variants truly are responsive to the drugs we have in hand, are proven to be safe, then are we really doing the right thing by not allowing them to have those drugs? Because we don't want to trust the results of cell-based systems when, in other cases, the cell-based systems have proven to be robust. So the cell-based systems don't answer every question. There are some concerns about how they're being used. But so far, in aggregate, they've proven to be very useful. So I believe people from the US CF Foundation and others are discussing with people at Health Canada, and hopefully and the CF Canada input as well will moderate or change the opinion here. But I don't know much more than that, but I'm aware of that problem, and hopefully it could be resolved for the patients I know that have contacted us about this issue. These are Canadian patients who have contacted about these issues. So I see the point about the video. Yeah, okay, it looks a little bit like Gattaca. I agree. Okay, I kind of bought that question and that comment, but that was the JH people. I'll have to reflect back to them. But they do say it's not all genetics, I will say in there. So I'll credit them with saying you got to consider the clinical part, something else. Okay, somebody go ahead, Alana. There's another question.
I used CFTR2 in the Toronto database last week in CF Clinic. Patient heterozygous for 2 very rare mutations. We need something to see if she might be a candidate for something.
Yes, and thank you for that question. We are proposing in the next round for CFTR2 to include functional testing results for the rare variants. I mean, there is an issue in the United States. Would this be sort of assuming a prescriptive kind of saying this works, but we would provide this in the context of experimental results and peer-reviewed results, and so that individual really rare variants would know their variant has been tested in the cell system, it was published, and there is a response. Then the physician can make the decision, because the physicians are allowed to make decisions with information in hand as to whether that patient might be eligible for the drug. And in some of these cases, we know the physician's gone ahead, prescribed ivacaftor, for example, and we have been told that they had, as expected, excellent results based on the functional assays done on this really rare variant. So yes, we're going to include it in CFTR2, and thanks for that question.
I think we passed the hour already, and I don't think there are other questions. So I will have to stop here. But thank you very much, Dr. Cutting, for this inspiring talk. I think inspiring, I hope, to our trainees, and certainly to some of the precision medicine initiatives that we have at the University of Alberta and our research institute. Unfortunately, we can't applaud you on the virtual world.
No, that's okay, but I appreciate it.
Imagine that we are all there.
The questions are excellent. I wish we had more time to answer. Yes, the arrows on the gene, you're right, should go both ways. Thank you, good question.
So just to say that Dr. Cutting has kindly agreed to give a second talk tomorrow during the pediatric grand rounds on pharmacogenomics related to CF. So please join us tomorrow morning at 8 o'clock Mountain Time for that second talk. Thank you.
Thank you. I'll see you tomorrow.
See you tomorrow.